Hello friends, today we are going to talk about the working of a pH meter. So before going into detail about the working of a pH meter, we should all know what is a pH meter, what it does. Okay, so pH meter is a device that measures the pH of a solution. Okay, and it is going to tell you the concentration, or you can say the hydrogen activity. Okay, and basically it has the key parts, some key parts. So the first of all, it has the solution that is going to be tested, a glass electrode, thin layer of silica glass containing metal salts, inside which we put or the potassium chloride, which is also known as electrolyte, and an internal electrode made from silver or silver chloride. Okay, and we have also the hydrogen ions formed in the test solution that basically interact with the outer surface of the glass, and the hydrogen ions formed in the potassium chloride solution interact with the inside surface of the glass. The meter, which is going to measure the difference in the voltage between the two sides of the glass and convert these potential difference into the pH meter reading, and the reference electrode that will act as a baseline. Okay, you can see here, this is the reference electrode here, so it will act as a baseline or reference for measurement. Okay, now when we dip the electrode into the solution, okay, so you can see there is a kind of activity which is outside and which is inside. These are called the hydrogen ions activity, which is going to move toward the outer surface of the electrode and replace some of the metal inside, while some of the metal ions basically move from the glass electrode into the solution. So this ion swapping process is basically known as ion exchange, and it is a key to how a glass electrode works. And ion swapping also takes place on the inner surface of the glass electrode from the solution. The two solutions on either side of the glass have a different acidity, so a different amount of ion swapping takes place. That means it is whether acidic or basic in nature. Okay, and this creates a difference of hydrogen ion activity on the two surfaces, which means a different amount of electric charge builds up on them. This charge difference, okay, which is approximately measured in terms of millivolt, or you can say a tiny difference, is called as potential difference. Typically a few tens or hundreds of millivolts appear between two sides of the glass, which you see on the the pH meter, okay, and which produces the difference between in the voltage between the silver electrode and the reference electrode, which is here. Okay, and that shows up on the meter. Although the meter is measuring the voltage, what pointer on the scale display actually shows up is a pH measurement. The bigger the difference, between the outside and inside, the bigger the difference in the hydrogen activity. If there is more hydrogen, if you see here, if there is more hydrogen activity in the solution, it is more acidic. Okay, and if the meter and this is a lower pH. In the same way, if there is a less hydrogen activity, it shows that the pH is more, which means the solution is alkaline. So you can see here the this is the glass membrane which is outside, and this is the porous membrane that allows the reference solution to slowly drain. Okay, and here basically the outer layer is also called as the hydrated gel layer. Okay, and here is my junction, okay, which will allow the the ions to percolate or pass through. Okay, and in case of the where the solution is neutral, in that case the potential difference applied will be approximately 0. Okay, now we have the another picture which you can see here. Okay, we have the we already talked about the reference electrode, and the two forms basically is the electrode pair or the combination, and this we already talked about, that is going to measure the potential difference. Okay, you can see there is another typical layout, okay, which we have the reference cell, the typically filled with the neutral solution, okay, like KCl, or you can also other use NaCl also. And the reference junction is basically porous ceramic or porous Teflon, which will allow the the ions to pass through. Okay, and then this is my probe solution in an acidic environment which has an excess of H plus ions. If we can refer to that, this will have the more hydrogen activity. Okay, in that case your solution will be acidic. Now we have some activity, that what is acidity. So you guys can comment in my comment section, okay, answer these questions. Okay, what is an acidity, and what does pH actually mean, and how does a pH meter work, and who invented the pH meter. So thanks for watching. Don't forget to like and subscribe.